Yeah, I mean, we're ready to go, right? And then if you can just announce into the microphone here.
Let's get started.
Yeah. All right.
Hey. Good morning.
Barry, the mic's not.
Can you guys hear me?
No.
It's not right.
It's not working.
It's not working. Mic supposed to work.
I don't know if I think mine's working. I don't know.
Hold on. Let's see. Mine. Wait, is this?
Oh yeah, there you go. Wait, so that one was off.
That's off. Wait, so then.
Oh wait, she hears you though.
Huh.
Alexis hears you. Why isn't it coming in the room? Maybe just speak loud.
Okay. Oh, that's but I can show.
Alexis says she can hear, but that
Yeah.
but I'm not.
No, why-
Is my mic working?
Yeah, your mic.
My
Yeah, I
mic's
can
working.
hear you. So maybe.
Do you want to borrow it?
Okay, I can borrow it. Yeah,
All
yeah.
right, yeah. Let's do that.
Okay, cool. Can you guys hear me? Yeah, I can. Good morning. Good morning. Let's get started. Welcome everyone to BME seminar series hosted by Department of Biomedical Engineering at the Columbia University. My name is Qi Wen, and I'm associate professor in the Department of Biomedical Engineering. Today, we continue to offer our lecture in a hybrid format with both virtual and live audience. Our speaker will present for approximately 40 to 45 minutes, and we'll leave for 15 minutes at the end for Q&A. For our virtual audience, throughout the presentation, please use Q&A feature at the bottom of your screen to submit questions. We'll try to answer as many questions as possible during the Q&A session at the end. For our live audience, you will have chance to ask your questions during Q&A session as well. So today we're very excited to have our newest member, our faculty member, the Professor Sanja Vickovic. Okay, I'm right. Okay, cool. Thank you. Sanja is a core faculty member of BME department and also the director of Technology Innovation Lab at the New York Genome Center. She holds joint appointment as assistant professor at Full Foundation School of Engineering and Applied Sciences and Herbert and Florence Ivan Institute for Cancer Dynamics at Columbia University. She is also Wellenberg Academy Fellow of Royal Swedish Academy of Science and the Royal Swedish Academy of Engineering. Wow, you're close to the Nobel Prize. Yeah, well, and Professor Vickovic is an experienced and accomplished engineer and the innovator of special transcriptomics technology called Visium, for which we are using, and now commercialized by 10x Genomics. Welcome, Sanja. It's yours.
Thank you for that kind introduction. And let me try to just start the presentation and share the screen and make sure everything is working. Okay, let's get started. So I'll probably be walking around the room because that light is a little bit getting in my eye, to be honest. Let's get started. So as introduced, I work a lot on spatial and single cell genomics and technology development that has to do with genomics. I have a background in engineering and did my PhD in genetics. And after which I kind of did a lot of computational biology at MIT, and I just recently joined Columbia and the New York Genome Center. So, with a lot of background in genomics and genetics, if you've ever analyzed data, you know how hard it is to analyze that data, given data sparsity and given kind of confounders in your data. And what do I mean by that is that you can take tissues from patients, and these tissues may degrade over time. You have to control for a lot of these parameters, and it's not just about how the tissue is collected, it's when it's collected, what other risk factors the patient was exposed to. So when I started in this field of technology development almost 15 years ago, we wanted to address one particular question, and that is, how can we develop genomics technologies with applicability to the clinics? So we asked one simple question: which of these technologies in the clinics is used every day and has potentially the largest impact? And we, of course, came about pathology. So what you're looking at in this picture is, tissue microarray, which this means that it's 50 circles. In these 50 circles, you have a representation tissue from 50 patients, and you're looking at 50 colorectal cancer patients. And then a pathologist will look at the pink and purple shading of these cells, and these have been stained with a particular dye. And based on this shading and the shape and size of these cells and how the cells are connected in the tissue, the pathologist, after years and years of training, can make a diagnosis. And then potentially, based again on the expertise of that particular pathologist, he can potentially say something about the grading of that cancer, which stage the disease is at. Again, sometimes this pathologist could get help from staining from a couple of markers. This basically means that, some of the diseases we've studied for many years. For example, in breast cancers, we have the HER2 marker. We can stain these tissues for a HER2 marker and see if any of the cells pop up as positive and how many cells pop up as positive and where these cells are located. However, it's actually extremely hard with this type of data to make de novo discoveries. And this is because back then, there was no high throughput way to analyze all the markers at the same time from these tissues. So we were dependent on these discoveries that were already made and staining the tissues with 1 to 2 markers. So we asked, what can we do about this problem? So we come up with an idea that seemed pretty simple at the time. It just took a long time to get it to work. Is that if we take any single tissue from the previous slide and we ask the question, well, what would happen if we split each of these tissues into a grid? And what would happen if we extract in each and single part of this grid some type of genome-wide information? So that means that you would either do DNA or RNA sequencing of parts of this tissue. And then I'm highlighting, for example, 2 parts of this tissue. They're orange and blue. And each part of this grid, we then term tissue voxels. So that's kind of a popular term in image processing terminology. And then we wanted to design an assay that does sequencing in each of these voxels, not just the orange and blue one, absolutely all the voxels in the tissue. I've noticed my mic turned off for a second. Still on? No. Where's the button? Nope. Nope. Okay. Maybe we need. Sorry for that. Give us a sec. No, it's not green, the light anymore. Oh. Battery dead. Yeah, battery's out. Okay. No.
Okay, Alessa says she can hear you at the podium. I don't know why.
Oh, she can hear.
But the room cannot hear it.
I know.
Is there a charger we can connect?
Did you hear the mics? I
tried. I turned all them on.
Okay.
Happen.
Yes. No. No.
And everyone is pretty low.
I think it probably because the source on the Zoom is different.
I mean, maybe if you stand here and speak loud enough for the room, that's Zoom here with that. You okay?
We,
Yeah, they mentioned somebody.
okay,
Yeah.
let's do like this. Turn. Can we turn the mic over there and I'll try to speak to the room?
Yeah,
So we
let's
don't
do
lose,
this.
so we don't lose too much time.
Let's do
Yeah.
that.
Okay. I
Okay.
hear her voice is loud.
Yeah.
I mean,
Maybe ask.
Yeah.
Okay.
Yeah.
Yeah.
So,
yeah, you guys brought it on fire.
okay, so we're doing this old school, actually giving a lecture and not standing behind the podium. Okay. So we start by saying, okay, what would happen if you could extract RNA or DNA sequencing information from each and single voxel or part of the grid from the tissue? So, many technologies back then were continuously talking about splitting tissue into pieces. Well, that's inherently very, very hard to do, and how to do that reproducibly. So instead of actually splitting the tissue into a grid, we first created a grid. This grid we call a spatially barcoded microarray, which you see on the upper left-hand corner. So we produced a grid as a matrix. Back then, when we published in 2016, this was a matrix of 33 times 35 spots.
Could you stand a little bit closer to the podium so we can feel it?
Okay. Right. And what was unique about this grid is that, we wanted to somehow encode with DNA where the positions on the grid are. So how we actually did that is that we ordered, if it's 33 times 35 matrix, that's approximately 1,000. Okay. So we ordered 1,000 different oligos from IDT. And each of these DNA oligos had an encoded ATGC sequence that was a little bit different from one another. That means that we deposited a very particular DNA sequence into position in the upper left corner that encoded position 1,1 in a grid. Then we took a little bit different DNA oligo and we sprayed it on position 1,2, just next to it. Then we took the third DNA oligo and we repeated this for position 1,3 in the Cartesian coordinate system. When we were done with producing the grid, what happened is that if we zoom into the middle, is that each of these spots carried its own DNA primers, okay? But it didn't carry a copy of one DNA primer. So each spot that was 100 micron in diameter carried probably 200 million copies of the same DNA primer. And that this repeated for all the positions or all the spatial spots in our grid. And then if we zoom in on the far right side, what we see is that how these DNA oligos that we bought from IDT were designed, that you had this spatial barcode, which is the ATCG sequence encoding the grid on your glass slide. It was preceded by an Illumina adapter, and then on the top, it had the polyadenylated detectors. So all of us that have been doing a lot of Illumina sequencing and genomics definitely recognize this as a typical RNA-seq library. After, what can you then do when you've deposited, millions and millions of oligos on a glass slide that are Illumina sequencing compatible? So after that, you actually take a tissue, you slice that tissue, you put it on your spatially barcoded microarray. Then you do H&E staining, which is the same type of staining that the pathologist was doing in that first slide when we were looking at the tissue microarrays. After you've stained your tissue, you image your tissue. So now you're recapitulating all this beautiful morphology and histology that every single pathologist every day looks at, but you're also encoding where your tissue is on the grid, which we later on use in the computational steps. But now the most important part comes in the lower panel. So instead of, as I said, splitting the tissue, we first created the grid. We place the tissue on top of that grid, and now we want to extract information from the tissue that it flows down onto the grid. So we do a bunch of very controlled enzymatic reactions. The mRNA molecules drop down and flow on our spatially barcoded microarray surface. They hybridize through the poly-A tail in the mRNA and the poly-DT capture sequence on our DNA oligos on our spatial grid. When that binding happens, now we can do a reverse transcription reaction on the surface. That means that we are using an enzyme to copy the mRNA information on top of our grid. When that step is done, basically you have a molecule that's very stable. You have positionally copied transcriptional information onto a solid surface, and now you can collect these spatially barcoded mRNA cDNA hybrids, and you can go into just regular paired end Illumina sequence. So if we want to visualize what these results mean, is that so before sequencing, you're dealing with the same information that currently we have in hospitals. So that's the regular H&E state. But after sequencing, you know how I said that pathologists can sometimes get help from one marker, right? The HER2 marker in breast cancer. Well, now in each of these spots or each of these tissue voxels, we are encoding thousands of genes worth of information, not just one. So we went from 1 to 10,000 in one single assay. So we published this paper in 2016. I was wrapping up grad school, and it took us 8 years to develop it. But then I was like, okay, we've talked so much about applying it in the clinics. Let's go try and find a cool project to work on. And the first thing we did is we asked, well, what would happen? And I don't know, many of you work maybe in the brain. Is that there's an institute, the Allen Brain Atlas Institute, who tried to do this exactly same thing by using one gene at a time staining. So, for example, the pathologist was using HER2. In the brain, we know a lot more targets than we know in breast cancer. So the Allen Institute went through this task of staining thousands and thousands and thousands of tissues with thousands and thousands and thousands of different antibodies to basically recapitulate spatial organization of all these 20,000 genes in the brain. What we can do with our technologies, what took them probably 10 years to do, we can now do in 2 days. So that was the level of kind of resolution and throughput we achieved with this method. To apply it, maybe I should just remove the cursor. I don't know if that will help. Remove that top part. Yeah. Hopefully. Well, I'll continue anyways. The mouse is down in this over there. Okay. Yeah. Okay. So apart from applying it in the mouse brain, when you work with technology, it's probably the first system you would go to because it's well studied. We know how the cells behave, how they're distributed, what the organization looks like, right? But then we tried to do a little bit harder problem. So can we apply this technology to study disease? So we paired with Professor Hamali Patnani, who's currently at Columbia University Medical School, and is also core faculty member at the New York Genome Center. So we asked whether we can study ALS. So to be honest, 2016, 17, I didn't even know what ALS was. But since then we've learned, and I've learned a lot about it. So ALS is motor neuron disease, which basically means, as the name says, is that your motor neurons die over time. What we know about ALS is that motor neurons that die over time are located in a very particular part of the spinal cord. So it either starts in the lumbar or the bulbar part of the spinal cord. Unfortunately, I would say in all cases, it's still fatal, 3 to 5 years after diagnosis. And what else we know about ALS is that, well, the more the motor neurons die, the more severe the symptom the patient has. And again, unfortunately, 3 to 5 years later, the patient dies. There are a couple of known genes that are implied in development of ALS, but 90% of all ALS cases are sporadic. What we also know about motor neuron loss in this particular part of the spinal cord is that it involves self-autonomous and non-autonomous processes. This basically means, well, we know the motor neurons die, but we know they die because of their environment, they just don't decide to die from nowhere, right? So I think this was a good question to ask with spatial transcriptomics. Because we know we had to measure gene expression spatially, and we knew we had to measure it over time as the disease progresses. In order to do this, we, in about 1 year worth of time, created so far still the largest spatial data set that exists. So we profiled over 1,900 tissue sections with ST, and these tissue sections you see as basically virtual stacks here on the left. And each of the dark spots basically denotes one of those spatial spots on the grid. Highlighted is the part of the spinal cord called the ventral cord. This is the part where the motor neurons reside and the motor neurons then die. On the right-hand side, you actually see overlay of the full data set we have. So that data set comprised in the end of 190,000 spatial spots originating from the spatial grids. We made a registration algorithm to register all these sections on top of each other in a common coordinate system, which you then see color-coded. Color-coded are different, very important kind of morphological areas typical for the spinal cord. Again, in orange is the ventral cord where the motor neurons reside before death. The important thing to note is that in each of these 190,000 spatial spots, we actually were looking simultaneously of expression of 12,000 different genes. So this is a typical result that you can get with spatial transcriptomics. So top panel is your wild type condition, okay? Bottom panel is your ALS condition. Each column is a different time point as the disease progresses. P30 means, mice have reached sexual maturity. We don't expect anything to happen. P70, mice are a little bit older. We're still not expecting much to happen, right? We see on the histology, the motor neurons are still there. Well, at P100, at P120, which is end-stage disease, basically we see how the motor neuron loss progresses from the histology. So we know also from the histology and from the symptoms, by tracking the mice are definitely exhibiting the disease, okay? What we then see, for example, if we look at Gfap expression, which is a gene that has been previously associated with ALS progression in your bottom panel, as the disease progresses, motor neuron loss occurs at P100, P120. We see a lot of overexpression of this particular gene involved in gliosis. But to be honest, we didn't study, we didn't set out to study ALS to look at one gene at a time. So what's very cool to look at with spatial transcriptomics is something we call gene expression programs or gene co-regulation programs. Which basically means that, there's a lot of different blue and red patterns, and they change over time, okay? So for example, some particular cell type in the upper left corner, it's very expressed, so very red in the dorsal cords. Well, as the disease progresses and we follow in the diagonal, this gene co-occurrence or this gene co-expression changes from the ventral cord all the way down to the white matter, okay? So that can imply either, the cells are not moving and they're communicating to each other, or actually cell populations are moving within the tissue as the disease progresses. In order to kind of be able to say something more about the cell populations, we integrated spatial transcriptomics with single nucleus, or you can do single cell RNA sequencing, which means we are taking published reference single cell or single nucleus data sets and marker genes of these cell populations, and we're mapping that in concordance to our gene expression programs from the previous slide. What happens is that we now know that on the left-hand side, there are some programs that are involved with neurons. On the right-hand side, we know some programs are involved with vascular cells or oligodenders, okay? Given, we created a very, very large data set, and we of course don't have time today to go through everything, Let me just show you two types of results you can expect. Top panel, we are looking at log fold changes. That's on your y-axis. And we're looking at log fold changes, what's overexpressed in your AOS condition versus the wild type condition. On the x-axis, you're again going through the different time points. So P 30, 70, 100, 120. So what happens, and then we're looking in the ventral horn where the motor neurons are dying. We see at P100, P120, a lot of things are up or down regulated because now the motor neurons have died. And of course, any gene that we find interesting, we can also go and confirm and validate using these a little bit older methods like immunofluorescence, right? Like the pathologists would still do. We can still do that. It just takes a lot of time to validate a lot of targets, right? But I think what was more interesting is that we see upregulation of some of these programs already at the time point P70. So at P70, the mice are not exhibiting symptoms and there's no motor neuron loss. But we see already there pre-symptoms that the mice are exhibiting ALS molecular biology. And I think this is maybe a little bit hard to understand, but let's try to go through it. So on the left-hand side, again, you're looking at some of these gene-gene combinations that popped out of the analysis. And again, you're looking at the time point P70. When the mice are not symptomatic, there's no motor neuron loss. What we see basically is upregulation of something that involves ThyroBP as a target and TREM2 as a target. The top two heat maps, wild type, bottom two heat maps, AOS condition. Okay, so we see co-regulation of these two genes. We also see that these two genes may be a little bit harder to see on the IFs, is that in the AOS condition, they are expressed just at the contact to the motor. So to be honest, this is probably one of the more interesting results from the study because we now have managed through a lot of wet lab work and a lot of computational work, deconvolve potential biomarkers and potential targets of disease prior to actual disease symptoms, and that we can actually show that these targets are expressed at the boundary to the motor. So they're directly impacting the motor neurons that are about to die in the next 10 days. And I think what's even more cool is that this study and these two targets got picked up by two pharma companies, and it's currently in clinical phase trials. So I think what this whole thing that took 10 years taught me is that, well, you have to be smart when you're developing technologies, but when you develop the version of the technology, you need to apply it to some type of actual clinical problem to see what are the excuse me, shortcomings of your technology and what can you do better. So I think we identified three things after the ILS study we wanted to work. One is that we wanted to automate the full process so that we're no longer students are pipetting and I'm not pipetting. Second is that we wanted to have multimodal signatures. So this means instead of only tracking RNA expression, we wanted to check, can we track protein expression? Can we track DNA expression? Second is that, well, in order to know that something is going on spatially in the systems, one modality is impacting the other, we have to actually somehow perturb our system. So instead of working a lot with CRISPR-Cas, another way you can perturb is asking what happens in your guts in the presence or absence of microbes. And then the third, and I think probably the most important part was, well, we were in the initial version of technology continuously dependent on these voxels. These voxels were 100 micron in diameter, means that you were capturing the same time simultaneously expression from 5, 10, 15 cells, right? So we wanted to increase that resolution to be cellular or subcellular resolution instead of these tissue voxels. So let's start off with spatial multi-omics. So spatial multi-omics is one of these let's combine modalities type of thing. So, we have talked a little bit today about IF staining or immunofluorescence staining. So that means you're buying an antibody. This antibody is modified with a certain fluorophore, a certain color, and you are basically incubating your tissue sections with these antibodies. Hopefully, this antibody binds to the correct antigen, and you're reading out basically the position and the expression of this binding by regular epifluorescence microscopy, let's say. There are limitations to that system, is that at the same time, you can only use a few colors because of the spectral overlap between these fluorophores. So let's say that at the same time, ideally, you can do 3 to 5 antibodies, So we wanted to overcome that so that, let's say that in your reaction, you can still pipette your 2 immunofluorescent labeled antibodies that are binding to 2 different antigens and have 2 different colors, like Cy3 and Cy5, okay? But at the same time, we're incubating our tissue sections with those antibodies. We'll actually incubate the tissue sections with DNA barcoded antibodies. So now instead of Cy3 and Cy5 labeling your 2 different antibody clones, you're going to have antibody clone 1, DNA barcode 1 bind to it. And you will have antibody clone 2 and another DNA barcode bound to the antibody. So after incubating your tissue sections with these antibodies, what happens is that the DNA barcoded antibody, again, finds hopefully its correct antigen. After the DNA barcode, we made and engineered a little poly tail. And now upon binding, this poly tail can bind to your spatial grid and spatial surface, same as your mRNA molecules that are polyadenylated can bind to the surface. And we extend from the surface. We're again copying now information of the barcode into some kind of a sequencing library. So what happens is that if we focus a little bit on your left-hand side, so we're looking at mouse spleen. In mouse spleen, you have canonical architecture of white and red polyp. Far left is where you actually see your immunofluorescence staining in Cy3 and Cy5, right? We're looking at beautiful blue and red parts of the tissue. What we see then on the right-hand side is that what is the actual sequencing signal from the DNA barcodes that were attached to the same antibodies in that same reaction. And so we see like quite a great correlation between, sequencing, antibody expression, and actual immunofluorescence that we're all very, very much used to. But we, of course, didn't develop this technology to only be able to, do 2 antibodies at a time. Because 2 antibodies we could already do in green immunofluorescence. Why we attach the DNA barcodes to the antibodies that we can multiplex. We're not dependent on spectral overlaps anymore. So what happened then on the right-hand side? Well, we can put in much more of these DNA barcoded antibodies. In fact, we put more than 50 antibodies at a time. I'm just for the purpose of screen showing you 6 different antibodies here. And what's other cool thing you can do then with the spatial multi-omics technologies is that if on the top panel, you're looking at your protein expression or your antibody-based expression of the proteins in your tissue, on the bottom panel, you can map that to actual mRNA expression. So you can actually then look at some biology and how much of the mRNA and protein signals actually correlate in cells in tissues. And as I said, along the way, we got very tired of pipetting these libraries and pipetting the reactions on the surface. So we took the robot we had in the lab and we coded absolutely everything to be done on that robot. So now the only thing my students do is place a section on the slide and put it on the robot. The robot does rest, right? We're focused then on actual technology development and actual computational biology later on. So this started off as a pet project because I was super interested in working in the gut and wanted to ask, well, what technologies are missing in the gut? Started working with this 2016, 17, 18, and back then there were only literally 2 papers ever published on spatial organization of the microbiome in your guts. And I think they were pretty cool. But I was, disappointed after reading them in terms of, I can only map 2 bacterial families, because again, we were dependent on immunofluorescence back then, and that's the only thing we could target in the microbiome. Okay, 2 fluorophores, 2 bacterial families. I could not know anything about the genera or the species or any lower level resolutions in the microbiome. And again, I could probably do this in very low throughput. So, you know, 1 tissue section probably takes me a month or 2 to optimize how to do. So we asked whether we can kind of re-engineer our spatial transcriptomics technology to do spatial host microbiome sequences. So what I mean by that is that, well, you know, the spatial surface, it's actually solid surface. It's a glass slide. On this glass slide, you have deposited your DNA oligos. Same as I explained, you know, these are these DNA spots in a grid. It would be a little bit too expensive to continuously buy different versions of the capture flow. So you remember in that first slide, I said, well, you have an Illumina adapter, you have a spatial barcode, then you have a poly-DP capture. And this poly-DP capture sequence is, you know, what you need to capture polyadenylated molecules. So if I would, for every project I want to do, have to change this poly-DP capture sequence and buy another 1,000 or 5,000 probes, we'll probably all go bankrupt pretty soon. So instead, what we did is we optimized how to enzymatically add or remove sequences on our spatial array. So in case of spatial host microbiome sequencing, we're simply adding a 16S capture probe. So if you're not familiar with 16S sequencing, that's the most variable part of the bacterial genomes. It has 4 different regions. We're currently targeting region 4. And based on sequencing of this region, we can probably deconvolve 97% or so of different bacterial species that have currently been mapped at all in the past 10 years. So the 16S sequence is basically used for identification of bacteria. So as I said, we have reverse engineered our own arrays to now capture both the polyadenylated transcripts, but also on top of that, the 16S variable sequences from the bacteria. Actually, I think that was equally as hard as coming up with a computational approach to deal with this data, because remember I said, you know, 2017, there was like 1 paper dealing with immunofluorescence and spatial organization of the gut. Well, you know, no one has actually done this before. So we started from scratch. So all the bioinformatics tools, we had to either build it ourselves or reapply what's been done and talk to a lot of people what they have tested to figure out what might work for us. So we actually assembled a new database of mouse microbiomes in the colons. We also made a new deep learning tool that maps these type of data and actually super efficiently at 98% accuracy can identify bacterial genera in your data. Which is, you know, if you look on the right-hand side, what that actually means that Kraken 2 is probably the most popular tool you would use for bacterial and identification of your samples. DL is our deep learning model. You know, they probably do a pretty good job when you're trying to classify bacteria at these higher levels, like phylum, class, order, or family. But when you actually try to see the differences bacteria, that's the genus level. Well, Kraken 2 cannot anymore map the data. So that's why I'm saying we put a lot of effort into developing these models. And then just for illustration purposes, so you know how the gut looks like. So you're looking at cross-section of a gut. You know, it's a little circle. On the outer side of the circle, there's actual tissue. It starts off on the outer side with muscles, and it moves to the epithelium in the middle. Then when the tissue ends, which is probably somewhere here, you have a mucus layer that contains a lot of different bacteria talking to your host cells. And it contains the pellet. And pellet can contain anything from bacteria, dead cells, fibers you couldn't process, and so So what we did is we have a computational tool to annotate column samples so that we actually know in which region of the column we are and in which region of the column are actual different bacteria. So this is a typical result. I'm showing the expression of a gene called actin. So it's an epithelial marker. As I said, the epithelial markers are on the inside of the tissue. You see a lot of red expression. Cool, that's where it should be. Like, okay, our method for the transcriptome is still working. We're still mapping the genes where we're supposed to map them. But then the cool part is that if we're looking at two different mouse models, on the left-hand side, you're looking at a germ-free mouse. Germ-free mice are the ones that don't have a microbiome, okay? We have depleted the microbiome out of the mice. The right-hand side, it's an SPF mouse, or you can just call it your most regular laboratory mouse. It has the microbiome, everything's functioning as it should. So what we see is that with our spatial host microbiome sequencing on the right-hand side, we know that the bacteria are in the pellet and in the nucleus where they actually should be. Again, fantastic. We know that the method is working. So now comes the fun part. So we're now focusing on this one particular very thin part of the tissue called the apex of the crypts. So the apex of the crypts are where the cells in the colon are most differentiated. Those are the epithelial cells that receive the signals from the bacteria. What we see in the apex of the crypt, we see expression, and I don't know if you're familiar, but on the top panel, you're looking at TSNE plots. This is basically dimensionality reduction plots of expression of certain bacteria. We see what we call 4 clusters. These 4 clusters represent 4 different parts of the column, going from the apex, upper mid, base. So we're going from where the bacteria talk to the host cells directly to actually how it goes down in the tissue, whether any of the actual bacteria penetrate down into the tissue, which has not been known so far. So we see in the apex of the crypt a certain expression of bacterial genera. And then what we did, again, computationally, we mapped it on published single cell and single nucleus RNA-Seq data to try to deconvolve which cell types are actually present there and which cell types are actually talking to the bacteria in the different regions. This is probably the toughest slide to understand, but I'll do my best. So you're looking at a heat map. A lot of red means a lot of positivity and a lot of expression and a good p-value, okay? Pretty simple. The heat map is split into 2. The upper part is the SPF mouse, so the wild-type mouse that actually has the bacteria. The bottom part is the GF mouse, the germ-free mouse that has no bacterial present. What you see on the rows is notations of different cell types from the previous slide. And then just next to the cell types, you see which actually bacteria are present and talking to these cell types. So what we see, for example, if we focus on the upper right corner, the flavonifractor bacterial genus is talking to different neurons, glia, enteroendocrine cells, fibroblasts, tough cells. Okay, let's focus on the tough cells. So the tough cells that are in the presence of bacterial genus flavonifractor are expressing actually, some different programs. And these gene programs are connected, for example, to toxoplasmosis, platelet activation, ENzyme receptor interactions, and so on. Okay, so that means tough cells in presence of flavonifractor express these gene expression programs. Okay, let's look at tough cells a little bit down when they're talking to Coppercoccus. When they're talking to Coppercoccus, they perform RNA secretion, a completely different program, right? We can go through this task all the way down, but what you see in the GF mouse is that, well, when there's presence of no bacteria, tough cells don't express these programs. So it's a direct way to measure crosstalk between bacteria and host cells for these gene activation programs. Let's just skip that. So, well, you remember I said, well, now let's stick to the third part we had to solve when, after the ALS study is, okay, let's go back to some technology development. We knew we needed to increase the resolution of our spatial grid. So a simple way to do that, if you're looking on a spatial grid from 2016 on the left and the big tissue voxel is like, well, let's just produce a much denser grid. I think that was probably the hardest engineering undertaking we took in the last 10 years, how to engineer very, very, very dense microarrays. But we were lucky that we started a collaboration actually with Illumina, who's the popular sequencing provider. But before they were a sequencing provider, they were actually a microarray provider, as most of these companies are these days. So we asked, how do we go from the left-hand side, which is our original 100 micron resolution array and 1,000 different oligos from IDT to making a very dense grid. So if you want to make a very dense grid, that means that you have to have a lot of DNA oligos and a lot of small spots. So we created a grid that's approximately, let's say, 2,000 oligos on the right and 800 oligos on the Y. Well, if you would buy these oligos from IDT, that's approximately 1.5, I think, million different oligos. And the IDT would be like the new Google, and we would, you know, not exist because it would probably cost us like $30 million to buy this. So we were, of course, never going to do that. But we kind of found this old technology called barcoded bead arrays. So actually, these beads can be pretty small. I think Illumina produces them from 0.5 to 5 microns. We just chose one that was pretty easy to read out for us. That's 2 micron resolution beads. But, you know, these beads have to carry their own unique oligo, which means we have to go through a process called bead pool generation. So we want to create a lot of different beads, but on each bead, you want to have millions of copies of the same oligo. Same as we had in the initial spatial spots, which turned out to be pretty hard. But in the end, how we decided to do that is, again, we chose to buy some oligos from IDT, but instead of buying 2 million oligos from IDT, we bought around 500 oligos from IDT. So what we did is we bought the first plate of oligos. In each of these 65 wells that you see on the left-hand side, we bought a different oligo, okay? And each of these 65 wells in the plate, we added beads. With some quick chemistry, these oligos attached to the beads. So what happened is that now in 65 wells, we had 65 different bead moieties, as we call them. In each of these wells, the beads all had the same oligo. What happens next is that we collect all these 65 wells worth of beads into one tube. Now in this tube, you have a mix of multiple copies of the 65 bead moieties. Then we go to our second IDT plate that has a second, basically, oligo. Again, 211 different wells. In these 211 different wells, we add the mixed bead moiety that has the 65 different beads in the tube. So we add in each of the 200 wells some of the 65 beads. We don't know which ones, some we add. Again, quick chemistry occurs, and what happens now is that after that reaction is done, and you have, again, collected from 211 wells all the beads in your tube, is that now your bead pool increased from 65 beads in a reaction to 65 times 211 different beads in your tube. Let's repeat that one more time. Again, we put now the second bead pool into the third IDT plate with the third oligo. Again, click chemistry happens. After we collect everything in the tube, now our tube has 65 times 211 times 211 different beads. And this is kind of then what actually one design on one bead looks like. So you have oligos from plate A, you have oligos from plate B, and you have oligos from plate C. On one bead, you have millions of copies of this one full spatial oligo. What you do next is that you take a silicon wafer. This basically means you take silicon, you acid etch it in particular little wells, and you make certain patterns on the wafer, which look like this. They're hexagonal. Each little well that we etched was 2.05 microns. If you load a 2-micron bead into a 2.0 micron well, means that only one bead per one well, right? There's no space for anything else, which was great. Then, you have loaded your bead pool on top of this glass slide, and you have to go through a decoding process. So before, we, in the old version arrays, we would buy the 1,000 oligos, and we know exactly like, oh, capillary goes to this oligo, and it transfers it to this position on the glass slide. There was no decoding. We know exactly where we deposited each of the 1,000 oligos. Well, now we just in a tube made synthetic oligos, right? So if we deposited them randomly on a surface, we have no clue where the oligos ended up, but we know exactly which oligos we used from the start. So I don't think I have time to guide you through how the decoding process works. I'll just tell you that, it's done at Illumina. Thousands of arrays are decoded by hybridization chemistry in about 3 hours. And what you get in the end is, again, your spatial array on a slide on which you can deposit tissues. How to do that is that, again, section, stain, image, do the reactions on the surface, collect the polyadenylated transcripts that they're binding to the poly-DT capture sequence, extend sequence, okay? So what ends up in, a 2016 technology, which is, still on the tissue boxes now, in 2020, 2021, we're doing HDSC or high definition spatial transcriptomics, which is after sequencing at 2-micron resolution. So you remember, how I said we could recapitulate what the Allen Brain Atlas did with the mouse brain in a couple of days. Now we're actually at the better resolution than the Allen Brain Atlas in a couple of days. We're not looking at voxels, we're looking at subcellular resolution. And then again, if we're looking at the cross section on the left-hand side of the mouse brain, this is the olfactory bulb, this is just the front of the brain. You're looking at the left and the right hemisphere, which should theoretically be symmetrical. And color coding is basically the color-coded beads on our HDSC slide by very canonical morphology of the brain. So the known kind of regions of the brain. But then on the right-hand side, what we're doing is if each row is just a name for the region of the brain, and on the x-axis is all the possible different cell types we have known are previously been associated with the brain. We can map, for example, 3, 5, and 6 in red. The little acronym means olfactory bulb inhibitory neuron 1, 2, and 3. We can map that particular cell type to a granular layer that's marked in red, which is this kind of middle part of the mouse. Again, we can finally go to our HER2 positive samples or breast cancer samples that we wanted to study 10 years earlier. And now we can finally, with HDSC, map what's going on in every single cell in the biopsy that's being collected. And again, map a lot of gene programs on top of that. I think now we're at the stage, so we went through, another round of 4 or 5 years of technology development, and now we're at the stage where we want to apply these technologies to answer some questions. Some that I can think about would be cool to try to address in different diseases is so saying, spatially, which cells are disrupted, which cell programs change in space and time. Can we actually map the function of these cells to disease states? Can we potentially come up with computational approaches and algorithms that will tell us which exact communications are disrupted between cells? What are the actual extrinsic versus intrinsic cell signals? And I would very much like to work on what's the effect of the drug in the system. Not sure we're there yet with the technologies, but something definitely to think about. I think my lab, going forward, at least in the next year, will focus on 3 main topics. One is how we can actually very smartly, statistically design spatial studies which means, well, if someone comes to you and says, I want to do a pilot, right? Well, I'm saying, okay, give me one section worth of data. This one section worth of data, we can now predict, oh, I think you should do so many individuals, so many tissue sections, so many replicates, right? To be able to have enough power in your study design. So this might seem trivial, but no one has actually done spatial power design before. So we were also kind of, reading 60, 70-year-old papers to try to figure out, does any of that apply to what we're doing? And it didn't, but it at least gave us some ideas where to start. The second thing is I will focus a lot on mapping these cell-cell interactions and communications in colorectal cancer and as colorectal cancer progresses, with a huge cohort in my lab in Sweden that will kind of provide these samples. And then I think, the third part is I have talked to a lot of people who have tried to use Victim. It's the technology that's now commercial by 10x Genomics. HDST will be as of Q1 next year. And I think everyone's stuck at the computation. They're stuck at understanding their data. They're also stuck because they don't have the computational resource that's needed to actually run this data. So with Professor Satija at NYU, we have started the collaboration of actually scalable computational design for spatial studies. With this, I will end to give us a little bit of time to try to chat. A lot of thanks to the funders and mentors. And, if you have any questions, please do reach out on my email.
So that was a wonderful talk, really. So much information. Thank you, Sanja, for that. And before we get to the Q&A, which actually I don't think we'll go to the virtual Q&A, but I just need to call everyone's attention to next week's seminar, which is on Friday, October 21st. So that will be, so it isn't actually next week, on Friday, October 21st, the virtual one. You guys do have some all day. So Professor Marianne Weisrecht from the University of Michigan, she presents optical diagnostics for pancreatic disease detection. And the rest of you will be here in the regular seminar, not live. So follow us on social media, and great to see you all. And right now, maybe we just have time. It's already noon, I think, but is there one, you could take maybe one question to.
I mean, I'm happy to stay as well and chat if
Okay,
we need to vacate the room. So.
All right, that sounds fine. So it's noon, so have a great week, everybody. See you next week here. And if you have a question for Sanja, just please come up and hang around.
Yeah, awesome. Thank you.